Get ready for a mind-blowing revelation. Scientists have taken a bold step by attaching a mushroom to a biohybrid robot body, and the results are nothing short of extraordinary. Imagine a world where robots don't just follow commands from silicon chips. They take cues from living tissue. That's exactly what these researchers achieved. The control signals originate from mycelia, the intricate underground networks supporting mushrooms. These networks possess an incredible ability to sense light, touch, chemicals, and temperature, and they communicate through tiny electrical pulses. Researchers from Cornell University Engineering cracked the code to interpret these pulses without harming the mushroom or distorting the signal. They translated these rhythms into digital commands, essentially teaching the robot how to move. But here's where it gets controversial. When the environment changes, the mycelia adapt their electrical patterns and the robot adjusts its movement accordingly. It's like having a living, breathing sensoromotor control system. The team at Cornell Engineering grew mycelium in small chambers equipped with electrodes and connected these chambers to two robots. The fungus became the conductor, setting the rhythm for the robot's movements. By merging mycelium with the robot's electronics, we've created a biohybrid machine that can sense and respond to its environment. In this case, we used light as the input. But the future holds even more exciting possibilities with chemical cues, explained Rob Shepard, a professor at Cornell Engineering. Mycelia, the unsung heroes of this story, offer a unique advantage over animal cells commonly used in biohybrid systems. They can handle rougher conditions, thriving in various humidity and temperature levels and growing on basic nutrients. This toughness makes them ideal for robots operating outdoors or in challenging environments where traditional sensors might fail. And this is the part most people miss. Mycelia form intricate, responsive networks. Their electrical activity isn't a steady tone, but rather a series of spikes and patterns, perfect for timing movements. This natural rhythm allows engineers to detect meaningful signals without getting bogged down in decoding every tiny variation. One of the biggest challenges was ensuring signal quality. Electrical noise from motors, wires, and even the air could drown out the biological spikes. The team developed a shielded interface to block vibrations and electromagnetic interference, allowing for clean recordings even while the robots were in motion. The software monitored the incoming signal in real time, detecting spike times and translating them into control commands. By focusing on rhythm rather than decoding a complex language, the system remained robust. The controller borrowed the concept of a central pattern generator, CPG, from biology, a circuit that produces rhythmic outputs to coordinate movement. The mycelial spike set the beat, and the controller mapped this beat to motor timing, allowing the robot to coordinate steps or wheel turns without micromanaging each joint. To demonstrate the versatility of their approach, the team built two distinct robots, a soft, spider-like walker and a small wheeled rover. Each robot housed a living mycelium sample connected to electrodes. When the fungus produced its natural spiking pattern, the robots moved. The mycelia were sensitive to ultraviolet light, and when UV light hit the chamber, the spiking pattern changed, causing the robots to adjust their movement by altering gait timing. Researchers also implemented an override mode, allowing them to pause or redirect the robot with an injected signal without damaging the tissue. This combination of biological autonomy and human supervision opens up a world of possibilities. Real-world applications are vast. Soil chemistry constantly changes, and sampling across large fields can be costly. Systems that respond locally to nutrient cues could revolutionize agriculture, applying fertilizer precisely where it's needed and reducing waste. Environmental monitoring also stands to benefit. Sensors often fail in wet, dirty, or variable conditions, but mycelia have evolved to thrive and react in such environments. By tapping into their sensitivity, soft robots could detect toxins or pathogens and adjust their paths without hard-coded rules for every scenario. Light is just the beginning. Mycelia excel as chemical sensors, reacting to nutrients, toxins, and microbial signals. Moving from light to chemical cues would open up direct links to soil health, pollutant detection, and other real-world triggers. A controller could listen for shifts in rhythm as the fungus responds to a molecule and translate that into a change in movement or task. This approach aligns with how fields and waterways behave, with chemistry changing in patches and pulses. A living sensor that tracks these changes in situ could enable robots to act locally without heavy computation. The significance of mycelium robots lies in their unique approach to machine control. Instead of treating sensors as simple accessories, this method integrates the sensor into the control loop itself. The living network doesn't just report data, it influences timing and action, offering a new philosophy for machine control. This project isn't just about controlling a robot, it's about forging a true connection with the living system. Once you hear the signal, you understand what's happening. Maybe that signal indicates stress and the robot visualizes the physical response, making the invisible visible, said Anand Mishra, lead author and research associate at the Organic Robotics Lab. While there's still work to be done before long-term field deployments, the key step has been taken. A living network can guide a machine beyond the confines of a laboratory, and the machine can respond without the setup collapsing. Click here to watch a video of this groundbreaking study from Cornell University. Insert link here. The full study was published in the prestigious journal Science Robotics. Insert link here. Don't miss out on future updates. Subscribe to our newsletter for engaging articles and exclusive content.